A very good day. This session we are going to learn about mechanism of action and mechanism of resistance with beta-lactams. Beta-lactams are mainly comprised of following antibiotics like penicillin, cephalosporins, imipenem, meropenem, aztreonam. In order to understand the mechanism of action of beta-lactams, we should know the normal bacterial cell wall synthesis. Bacterial cell wall synthesis takes place in various steps. The usual step starts with the glycan chain synthesis, and there will be sheets of glycan chains which comprises of N-acetylmuramic acid alternating with N-acetylglucosamine. Now these glycan sheets are well connected by a peptide link, and this peptide link bridge or cross-linking is mainly carried out by penicillin binding proteins. Penicillin binding proteins mainly carries out two types of reaction. One is transpeptidation and carboxypeptidation. So understanding of the peptide linkage to these glycan chain is very important final step in the synthesis of cell wall. Now mechanism of action of beta-lactams. The beta-lactam structure have two rings. One is beta-lactam ring, which is the active component of the drug, and the second one is thiazolidine ring. So knowledge of beta-lactam ring is very important because that's the one which mainly takes part in the mechanism of action. So beta-lactam basically goes and bind to the penicillin binding proteins. Once they bind to penicillin binding proteins, these enzymes becomes ineffective, and that's why there would be no peptide cross-linking. Hence, beta-lactams inhibit both the process, that is transpeptidation and carboxypeptidation. Mechanism of resistance. The first mechanism of resistance which I am going to discuss is efflux pump. Now, some of the bacterial cell wall, they have active efflux pump, and whenever beta-lactam enters the cell wall, this active efflux pumps will try to pump it out. And this type of resistance is mainly seen with bacteria like Pseudomonas E. coli. Another mechanism of resistance would be production of beta-lactamases. Now, beta-lactamases are the enzymes which are produced in the cell wall of the bacteria, and whenever penicillin or any beta-lactam enters into the cell wall, these beta-lactamases will cleave the beta-lactam ring. After the cleavage of beta-lactam ring, as it opens up and it becomes ineffective, and thus it makes penicillin ineffective. And the major bacteria which develops resistance by these mechanisms include Staphylococcus and Pseudomonas. Hence, against such organisms, drugs like methicillin, oxacillin, cloxacillin, nafcillin are very effective, especially against Staphylococcus aureus. The third type of mechanism of resistance you would see is alteration or mutation in the penicillin binding proteins. If there is mutation or alteration in these penicillin binding proteins, then the beta-lactams are unable to bind to these proteins. And as they are not able to bind to this PBP, that would lead to resistance, natural resistance of this penicillin. Now currently, certain Staphylococcus, they are developing such type of resistance, and they are called as MRSA, which is methicillin resistant Staph aureus. So drugs like methicillin, oxacillin, cloxacillin are not effective in such species of Staph aureus. So that finishes the mechanism of action and mechanism of resistance to penicillin. Thank you. I'll keep updating my videos. Please subscribe to my channel. Thank you very much.